Welcome to this video on considerations for collecting and using natural history data that are fit for use in the regulatory setting. A natural history study can be helpful for planning drug development. However, the natural history of rare diseases is often poorly understood. A natural history study is a pre-planned observational or non-interventional study intended to track and gain understanding of how a disease develops over time. Information obtained from a natural history study can assist in drug development at every stage, from drug discovery to the design of clinical studies intended to support a drug's marketing approval. When collecting natural history information, various potential uses of the data should be considered, because data collected initially for one purpose, for example, to obtain comprehensive clinical information on patients with a particular disease, may not be fit for use for another purpose. There are several ways that a natural history study can inform the design of a clinical trial. A natural history study can help define the patient population for a trial. Information about disease subtypes, signs and symptoms, and rates and patterns of progression are useful in informing trial design. It can help identify or develop biomarkers that can be diagnostic, prognostic, or predictive of treatment response and durability. It can also be useful in guiding participant recruitment and dose selection. It may also identify aspects of a disease that are appropriate for endpoint assessments that can be used in future clinical trials. And it may serve as an external control in a clinical study. However, its use in this context requires careful assessment and planning and will be discussed later in this video. When considering the use of natural history study data, sponsors should note the following. Natural history studies should have well-defined, carefully documented protocols completed before initiation of the study, and any changes over time should be documented. If the study is intended to provide essential support for a drug application, sponsors must ensure that they are able to submit patient-level data to FDA when required. Sponsors must also ensure that such data meets applicable FDA data standards. Before using natural history data in a study intended to support regulatory decision-making by FDA, sponsors should consider whether the data are fit for use by assessing the data's relevance and reliability. Relevance includes the availability of key study variables, such as exposures, outcomes, and covariance, and sufficient numbers of representative patients for the study. Reliability refers to accuracy, completeness, and traceability. Sponsors should consult FDA regarding the appropriateness of using natural history data that are intended to provide evidence in support of a regulatory decision. In certain situations, a natural history study may serve as an external control. The source population for the external control arm should be as comparable to the treatment arm population as possible. Given that externally controlled trials do not involve randomization of the study population, there are sources of bias which must be considered. Reducing the potential for bias in externally controlled trials is best addressed during the design phase. Results from the clinical investigations using an external control are most interpretable when certain criteria are met. These include when the treatment effect of the test drug is anticipated to be large, is measured in a manner that reasonably minimizes and manages bias, can be objectively measured, and has a strong temporal association with administration of the test drug. For more information on FDA guidance documents related to externally controlled trials and natural history studies, go to the guidance tile on the Accelerating Rare Disease Cures or ARC program website. Important design considerations should be discussed with FDA early in drug development, particularly if data from the natural history study are expected to be submitted in a marketing application. For more information about different types of meetings that FDA offers, please see the Leader 3D video, Challenges, Strategies, and Regulatory Considerations for the Design of Rare Disease Clinical Trials. Additional information can be found on the FDA Leader 3D video landing page. This video was created as part of CDER's Accelerating Rare Disease Cures program. For more info on relevant guidance documents and the ARC program, please visit www.fda.gov/drugs/CDER-ARC.